Short and sweet. First up is Myron Child. Myron came to our program after a series of full-time research positions that ultimately allowed him to migrate intellectually from physics and astrophysics, which was his undergraduate degree at UC Berkeley, to, I would say, biophysics, to molecular genetics, to evolutionary genetics, something like that. Yeah, so there was a logical route between those somewhat distant endpoints. And Myron is going to tell us about some ongoing work that's been both interesting and challenging in different ways, looking at potential incompatibilities within Drosophila melanogaster and what they can tell us about the earliest stages of reproductive isolation. All right.
All right. Thank you for that introduction. Make sure that's working. Okay. Yeah, super excited to be here to get to talk to you for the first time about the work that I've been doing for the last few years in the Pool Lab. And so, as John mentioned, I'm going to be talking about the phenotypic and genetic basis of reproductive isolation between wild-derived fruit fly populations. So, as John alluded, I have come from a very interesting past, but my general questions that I'm interested in evolution at this point are, how is it that one population of sort of interbreeding fertile individuals becomes two different unique species, and what are the processes that lead to that? And so obviously that initially starts with a single population, but that the individuals and the genomes in that population are subject to forces like genetic drift, adaptation, and forces of sexual selection between sexually interacting organisms. And as those forces continue to act on the genomes, you'll start getting variation developing in a population that can start leading to scenarios where you have individuals with genetic differentiation between them that are becoming more and more incompatible with each other due to those differences. But those same forces that led those in the first place are still around and interacting and feeding back into those individuals. And so you have these very early stages of kind of isolation that can then become full-on reproductive isolation between subsets of a population that we then might start calling different species. So a lot of interesting work has been done in turn the laser. A lot of interesting work has been done on the sort of right side of this little display here in populations that are really well-documented species at this point, very isolated and diverged from each other, and the phenotypes of their isolation are easier to notice as well. But comparatively less has been done to study these really early initial stages on how those this isolation and those barriers arise in the first place and how they carry forward. So that's really kind of the place that I'm interested in studying. And so turns out we have a pretty cool system to do that with in fruit flies. So we're looking at the species Drosophila melanogaster that everybody uses for all your fun genetic stuff. But it turns out Drosophila actually exists outside in the wild. The Drosophila melanogaster is originally from southern Africa and has since expanded, and now you can find them in most of the world. So we have populations from a ton of different places across the world. So if we take two populations, one from the ancestral range, so in this case, we're looking in Zambia, and one from more cosmopolitan populations, say France, these populations are only diverged from each other about 13,000 years, but they do have this pretty significant barrier between them, being the Saharan Desert in Africa. So if you take flies from each of these populations and cross them to each other, they're still the same species, so maybe not entirely surprising that the hybrids between them appear to be viable and normal. We don't see any inviability or sterility in either of these sexes, which is basically what you would expect. However, if we take these hybrids and cross them to each other to make an F1 generation, or to make F2s from them, what we actually find is that a significant proportion of the males from these hybrids are actually completely sterile, while the females are still, again, totally fertile and viable. So this is a very interesting system, because we notice the early stages of where we're just seeing some males being sterile, and only sort of at the second generation, which implicates more interesting genetics. So this is the kind of system that we're working with here. But what also makes this very powerful is that we don't have just a single strain from each of these populations, right? There's a ton of flies in each of these locations. So we can actually look at several different lines compared to each other. So we could take, say, 5 different genetic lines from France and 5 different genetic lines from Zambia, cross them to each other, and see what kind of results we get. So this is work that's actually been done and published previously by a grad student in the lab, Matt Lawler. And so the numbers that you're looking at here are, if we did a cross and got to F2s, what fraction of those F2 males were completely sterile? And the 2 different numbers that you're seeing in each of these boxes represent whether the female that generated that cross came from France or from Zambia. So they're just reciprocal cross directions of the same strains. So a few things that are interesting to notice here. First is that we see some levels that are very significantly higher than what we get if you cross any random France strain to any random France strain, or same with Zambia. Order of magnitude higher in several cases. The other thing that you'll notice is that there's a lot of variability. Some lines we never see sterility rates that reach the statistically significant threshold. Other cases we see it in multiple lines, regardless of what it's crossed to. And then the other interesting thing to note is that there's some particular cross-directional effects. So you'll see some like this, where in both directions of that cross, we see sterility rates that are significantly higher than we expect. In other cases, we see it really only in one direction. And we see that in both directions, depending on which cross that we're looking at. So we have these really interesting genetics. There's a lot of variability that really sets us up to be a cool system to study. But this is way too many crosses to study. So to try and look at these further, we focus on just a subset of them. So the ones highlighted in yellow here are the ones that we are focusing on going forward. Just basically picking a subset that tries to capture a lot of the different phenomena that we're seeing here. So 2 of them where we see significant or really high levels in both directions, and then 2 when we only see it in one of the cross directions. And another interesting thing to kind of set up what's interesting in the system is that, again, we only see the sterility in those males, which is a phenomenon that is observed across a lot of biology when you are interested in studying those very early stages of speciation. So this is often called Haldane's rule. And it's basically just an observation that when you see reproductive barriers developing between 2 populations, you're almost always going to see those representing first in the heterogametic sex, which in this case means whichever sex has the 2 different sex chromosomes, which in the case of Drosophila is the male. So this is the XY. And we see this manifesting in sterility in those males. And there's a lot of different causes for that that are interesting and can be relevant in different cases. But this is basically just a sign that we are indeed looking at an interesting place for that early stage of speciation. So summarizing a lot of that information, we don't see any effects in the F1s, which suggests to us that there's multiple loci with recessive alleles involved in the sterility here. Otherwise, we should have seen something in these just heterozygous F1s. The sterility is incomplete. So we see total sterility in the males, but only around 10% of the males end up being sterile when we see it. And then we only see it in the males too, which suggests those early stages of isolation that we're interested in. I didn't show the data here for brevity, but Matt has also done back crosses where we generate these F1s and then back cross them to both of the parental strains. And we actually don't see any elevated sterility in those back crosses either, which suggests to us, at least in those cases where there's some kind of cross-directional dependent effects, that there should be more than 2 loci causing that. Otherwise, we should see that in the back crosses. So some interesting genetics definitely underlying that are a little more complicated. And then the fact that there's a large variability between the crosses suggests that there are potentially fewer but larger effect loci underlying this. If the sterility was some sort of very multigenic additive effect, you would expect to see, sort of middling rates across all of them, not these stark drastic contrasts between sterility rates, even just swapping cross direction. So suggesting some interesting genetics going on there as well. So sort of what we knew going into this. And what I wanted to figure out is what is causing the hybrid sterility. And so there's two components of that. There's the phenotypes of how is it actually presenting, and what are the genetic, with underlying genetic bases for that. So the test that we do for sterility, it's really high tech. We just give a male a bunch of opportunities to mate with females. And if they never end up reproducing with the females, we call them sterile. So it's a really conservative assay, but it doesn't tell us what exactly is causing the sterility in the first place. And then using that information, hopefully we can tell something about the actual genetic components of those. So the first thing we looked at, not knowing what the phenotype is for the sterility, is that, well, maybe the males are just not mating with the females. That would be a quick way to prevent you from producing offspring. So we did a bunch of behavioral experiments, filming courtship and examining the literal sort of song and dance that fruit flies do in their courtship displays. And it turns out, long story short, that we don't really see the barriers being behavioral. The males pretty much mate at the same rates you would expect. They don't seem to have a terribly difficult time courting the females. So going from there, we said, well, let's see if the males are actually transferring sperm to be able to fertilize the females with. So we can, after these matings, you can basically, as soon as it's finished, dissect the female's reproductive tract and examine it for the presence of sperm. And the fruit flies actually, the females are able to store sperm for several weeks after the mating. So it's usually pretty easy to find sperm present in females if it's been transferred to them. And again, without going into all of the testing, we did this a lot. Don't ever really see the sperm getting transferred to the females. So it suggests to us that something is going on the male side that is causing this. So the first thing started dissecting males. And one of the obvious things we noticed is that many of the males end up having very severely reduced. So if you look at this little schematic up here, the Drosophila testis is really long, which is needed to produce the very long sperm that Drosophila have. I think it's around a 1.7 millimeters or something. It's something you could probably see with the naked eye if it was thick enough. So they're surprisingly really long. And so they have these very long testes. As you can see, an example of a fertile male here, where these are very long. The sperm develops in here, and then once it is fully developed and motile, it gets sort of stored here as mature sperm. And if we look at these sterile males, these are examples where the size is severely reduced to a point where spermatogenesis is not even able to proceed in here. We also see cases where it's very asymmetric in the reduction. So there's maybe some sort of stochasticity in how that reduction is playing out phenotypically. But we only see this in some of the males. This is not all of the males that end up looking like this. Some of them we see fairly normal length looking testes, but if you dissect the testes, we don't actually see motile sperm within them. So in the case here, the testes are so short that they're not able to produce the sperm. But in a case like this, nothing about the testes should be preventing them from producing sperm, but we still only see these sort of midway through spermatogenic unindividualized sperm bundles. So spermatogenesis is proceeding to some degree, but it's not getting all the way there, so we don't actually see motile sperm. So some of them are not producing sperm in that way. But this is also not the only other phenotype that we see. So it turns out some of these sterile males actually do have motile sperm. So that's sort of an example of that picture I was just showing, where you see just these kind of bundles and some of these early stages of the spermatogenesis. We see some cases like this where we have, you can see here, there's like a few motile sperm moving in here, but otherwise you're seeing mostly these sort of unindividualized bundles of sperm. So spermatogenesis proceeding to some degree, but not fully. So obviously something is going on here. Here's another male that is also sterile with a lot of motile sperm that you can see are very long and coiled and moving along around here like you would expect. So we see a lot of different phenotypes. Summarizing that a little bit better here, what you're looking at is each of these columns here is one of the different 6 crosses that we looked at. You'll notice the first 2 are just the reciprocal crosses of each other. These 2 are reciprocal crosses of each other. And then the 2 one-directional crosses that we looked at. And some of the things to draw your attention to here is, one, we see pretty different phenotypes between the crosses. So in this case, we see an instance here of a fairly severely reduced testis, but most of them appear to be very normal. and have motile sperm at a really high rate. We see another case like this where majority of the testes have some sort of reduction in their size and are not producing motile sperm. But there's also cases within here where they have normal length testes, but sperm is not being produced as well. The other interesting point to note here is that, again, we see very different phenotypes between these two, even though they're just the reciprocal crosses of each other. So the same strains used to make this cross, just in different directions. So summarizing that information a little bit, the sort of phenotypic basis of sterility as we're finding it so far is that in all of the crosses we've looked at, none of them seem to be transferring sperm. But in some cases, that is because the males are not producing sperm. And that can either be from testes reduced in size to the point they're not able to produce sperm, or in other cases, some sort of error in spermatogenesis that we're not quite sure of yet. In other cases, the males have totally normal testes and motile sperm, and they're just not transferring it to the females for some reason. So there's a lot of variability between these, and even within the same strains used to make a cross. There we go. Yes, so there's several different ways we could go to continue to look at some of these phenotypes as well, which we're interested in. So in the case, for example, of males that have motile sperm but just aren't transferring it, what is going on there? It could be something wrong with the genital morphology of the males. If they're not very accurately aligned with the female genital morphology, it can lead to problems in them being able to transfer. There's some good evidence that male genital morphology is subject to pretty strong sexual selection. It could also be some sort of muscular control or problem with the males internally. So there's different ways that we can look at that that would require more careful examination of those. There's also the case where the males are not producing sperm and have reduced testes. You can look at things like the sort of chicken and egg problem of if the testes are reduced in size and thus preventing spermatogenesis, or there's actually evidence to show that when spermatogenesis is experimentally halted in males, they can lead to atrophying of the testes. So you could have an error of spermatogenesis that's leading to reduction in testis size. And so there's different ways that we are wanting to look at that too, sort of dissecting males before the atrophy could happen and see if we see reductions and like that. So a lot of different interesting phenotypic information. But I also want to talk about the genetic basis of these incompatibilities. So in order to look at this, we have all of these hybrid F2 males, both sterile and fertile, that we can look at. And we can actually sequence hundreds of genomes from these flies. We have whole genome sequences from each of these, which is luckily a lot faster and cheaper now. So we can sequence all of these flies. And then because we know the strains and the crosses that generated each of these hybrids, we can take each locus along the genome and assign it an ancestry as either being having both copies at a locus from one parent, the other parent, or being heterozygous. And so we can go through and call that for each locus that we want to look at on a genome. And then we can go and take any pair of loci that we want to look at, scanning across the genome, and see which of those correlate with having mixed ancestry and sterility, since we know we don't see it in non-mixed ancestry, right? It's only in these hybrids. So this is the general process of how we are going to, how we're looking at these. And a lot of this work is also spearheaded by Matt Lawler again. And so this is kind of an example of what we would see in an hypothetical F2 male. So we're only really having about a generation of recombination to look at here. So you might have some areas where most of the chromosome is heterozygous if you get a copy from each population, but through some recombination event, you get part of it that swapped in a Zambia ancestry. And so now you have homozygosity, or you have homozygosity at that location. You can see the opposite direction too, depending on which chromosome you get from each parent. So this is generally what we were sort of expecting. And we started to look at all of these hybrid F2s, and we're getting some confusing results. We were seeing things that were suggesting maybe we were assigning the ancestry incorrectly. So Matt, after spending a lot of time bashing his head into the keyboard to make this work, turns out looks like we're actually, the ancestry calling we were doing was pretty accurate, but we were just seeing some surprising results in our F2s. So here's an example of an F2 that we found here. Two of the things to notice here are that, one, we see a lot more switches between ancestry than you should expect from an F2 male. Some of this could be due to some kind of error, or if you're if you're not sort of penalizing swapping ancestries in your ancestry calling method in certain ways, but we're seeing way more than we should expect. The other thing is that we are seeing, you'll notice, both homozygous ancestries in a single chromosome, which is not something you should be getting on a fly with only a generation of recombination. So using this, seeing this, we were started becoming a bit concerned. So this takes us to reproducibility. The first step that we wanted to do seeing this was to basically confirm the genetics of the lines that we were looking at. So here are some ancestry calling from what should just be basically the parental strains used to generate these lines. And whereas ancestry is being called, you can either do it based off of a single specific sequence or sort of panel of sequences using individuals from multiple strains from the same population. But we're seeing some concerning things here as well. This led us to be a bit concerned that the genetics of these strains might not be exactly what we're expecting them to be. So from there, we went through and resequenced every one of the lines that we were using to generate these hybrids. And we can use the sequences we collect from each of these strains to compare to old sequencing runs we have from these strains when they were first published, and other points in the lab as we've worked with them throughout the years, and basically make sure the genetics are behaving and appearing as we expect them to, given the kind of experiments we need to be doing with them. And then using each of those, we can basically compare for sequence mismatch and similarity. They should match to things that are the same sequence from the past, and they should mismatch other lines that are not them. So I'm going to show you some just a few examples basically of the scanning that we did, looking at all of the different lines that we had. So one of the metrics we can do this with is genomic distance. We basically just look at the entire genome of a fly and see how similar it is to the genome of another fly, essentially. And so what we have here is, say we have 3 different versions of a strain from France line 1. We might have one that was a older version, and then maybe 2 working copies that we've expanded to do experiments with. We have another France line 2 here, where maybe we have multiple versions that we've expanded and worked with, and we sequenced these another time in the past. And then we can compare each of these versions of those lines to the sort of original sequence for each of those strains that we had. And we can also compare them to another line that is none of them. So a lot of things to look at here, but I just want to draw your attention to a couple of examples. The first is that what we clearly see is that some of these strains don't match their old sequences. So if you look at these 3 lines here, all of these should be the same France line 1. Only one of them has a strong similarity matching to the old France line sequence that we had. The other 2 do not match that, which is not good. There's another example of this different France line. All of those should be matching to this old France line 2 here. We don't see any of them very clearly matching, which is also bad. The kind of opposite of that is that we see some strains that are matching things that they should not. So this version of this France strain actually turns out to be basically a swapping of a copy from a different line, matches this one. This one also appears to be very similar to a completely different France line. So there was very clearly some line contamination. The good news is that we have multiple copies of these. So not all of our strains are contaminated, but of course, just so happens that the copies that we used to generate a lot of these experiments were the ones that ended up having the contamination, which is always how that goes. So where does that leave us with the data? Well, it means I need to recollect a bunch of it. So there are some things that'll save us a little bit here. First is that there's a really strong reason to think that the phenotypes that we're noticing here are actually real and reproducible. One of the reasons is that the other strains that could possibly be contaminating some of our lines are containing a lot, if not all of the same alleles, just in different kind of combinations with each other. So the alleles that might be leading to the sterility traits that we're seeing are likely still around and present. What the contamination would make it difficult to do is the actual mapping of those genes, because you're getting a bunch of noise thrown into the genome that's just going to really severely reduce your power to detect those specific correlations with the genes that you want. The other thing is that since we were specifically screening these F2 males that we had for specific phenotypes of sterility, we're essentially biasing the samples that we're collecting in the sterile case to those that have the incompatibilities and thus likely to have those genes in the first place. So there will certainly be some slight alterations in this. Like, for example, you might expect it wouldn't be unreasonable for this individual to not actually appear in this cross if we do it again. Some of these proportions to shift a little bit. But the stark contrast we're seeing between these crosses is likely still pretty real. So working on regenerating a lot of this data going forward. Luckily, we know at this point some things that we care more to look at that seem important, some behavioral things we can probably study less now that it doesn't seem to be quite as relevant. And we can do kind of focus our attention on what parts of the data we want to take going forward. So it shouldn't be as painful to regenerate this as it was to discover all the things we want to look at in the first place. So with that, I just want to thank everyone in the Pool Lab. Matt, who did a lot of the groundwork on this and a lot of the work on the mapping. And then Jamie Freeman, our lab manager, who worked helping with the confirming the strains and the potential contamination, as well as a bunch of things with all of the fly work. And I just want to thank my thesis committee as well. And with that, I can take any questions. Yeah.
I just wanted to kind of calibrate on the genomic distances between your strains. What would you expect if you, for example, compared the Zambia strain with the France? Is that much higher than what you're seeing, or is it about comparable to that, like 0.05, 0.05?
Yeah. Yeah. So the scales here are just looking at France lines. But if you compare France and Zambia, sorry, the question was, what is the kind of scale of genomic distance that we expect? How different is the Zambia line from these France lines? And you can overall pretty clearly distinguish France from Zambia just in terms of their genetic distance overall. There's a little bit of nuance in there in that Zambian strains have more genetic variation overall, especially between each of the strains. So it wouldn't be unreasonable to see the same amount of genetic variance between two different Zambia strains as you would a Zambia strain and a France strain. But two France strains would probably look pretty similar to each other. So I didn't go into all that nuance here, but yes, there's in general more genetic variation in the Zambia strains, and they're pretty usually distinctly clear in terms of that genetic distance from the France strains overall. Yeah.
I love the rigor and reproducibility section. You are not alone in this.
Thank you. Good.
So, but I guess my question is maybe more hypothetical. So, given that all of your mutants seem to all point to some sort of sperm process, right?
Mhm.
Different ones. How does this relate to the kind of way you set this up, which is Haldane's rule, which depends on which chromosome you have, right?
Right.
Given that some species, the bottleneck seems to be with the females and not with the males.
Yeah. That's a good question. The question is about Haldane's rule and male versus females, where you sometimes see barriers in the females. Haldane's rule is interesting. There's different causes that people will cite for leading to Haldane's rule. Some of them apply specifically to being male rather than being heterogametic sex. Some of them apply to specific things about whether you're heterozygous on your sex chromosome. So there's a little bit of nuance there. So it's a sort of commonly cited, noted phenomena that you see it in the heterogametic sex, which to us would suggest if we're at the early stages, we should likely be seeing this in the males first. You'll still see those barriers in the females, obviously, but you usually you need a little bit more genetic distance between populations before you start seeing it in the non-heterogametic sex. But there's certainly weird counter examples to that are interesting and perhaps point at different causes of Haldane's rule that might eventually lead to the point where the rule itself is not very good and the explanations for it are probably the more relevant thing. I don't know if that answers your question, pontificating. Yeah.
Because if you said that you get the males and the females and you back cross and they're completely fertile without any problems, right? With the original population.
Yeah.
That means, at least in my mind, that the problem is not with one sex, but with interaction between that individual or whatever sex that we're doing to the other individual to the other sex. Because if you get this individual or this population, whatever, and get to another population, it's completely fertile, right? So might be a problem, not that they're not producing the sperm, because even if they produce like 100 or less than the total amount of sperm, one sperm is enough to fertilize the female. So maybe something in the female is stealing the sperm from the male. Meaning, it's important that the pair that you're doing and not the individual that you are analyzing.
Yes. So the question is there something with the female that we're mating these sterile males to that could be leading us to not seeing sperm in them rather than something with the sperm? It is interesting. There is actually evidence to suggest that females, their reproductive tracts can be spermicidal to some degree. It is surprising that you would see it that fast, because you can basically within a couple minutes be looking inside the female's reproductive tract after the mating has finished. It's a pretty quick time scale to be able to see something of that's that scale. It is also we consistently use a single strain to mate these two in the dissection test to try and avoid kind of strain different effects. So it would have had to have been basically a strain that was doing it for all of the individuals that had sperm, which is not impossible. It seems a little bit, I don't know, maybe a bit harder to justify. The other thing is I think you would still be able to see the sperm. It just would likely be, sort of inactivated or immotile to some degree. I don't know if you'd completely see all of that material being, disappearing.
If it's a person, yeah, you would definitely be able to see something there. If it's not, maybe I don't know. But how can you explain that you get the same population and that cross to another population, they're completely fertile? So the problem is not with them here, but with the pairwise comparisons that you're doing.
Yeah. So to me, that the question is about trying to understand why you have a strain that is sterile in one cross and not another.
Yeah.
To me, that suggests that there's different loci causing it that are you need multiple loci in a specific combination to lead to that. And like I said, a lot of the alleles are present just in different combinations between these strains. So, I mean, to me, it suggests that you might have two of the four loci in this strain, and if you cross it to this strain, this only has one of the four loci needed, one of the other four, and this one has the other two that you need. So you have to have all of those together or something like that. So it suggests, I would think to me that there's just different sets of alleles that are causing these that you're only getting in certain combinations of strains to each other. I don't know if that. Right.
But that's before the formation of the zygote, right? So it's something intrinsic of that male that's not producing the sperms. So why it's not producing the sperms? Doesn't matter if you put that female and it's crossed or not. It's not producing the sperms here. But when you put the same male with another female, it starts producing the sperm.
No. Okay. So yeah. So the phenotyping that we're doing, looking at the presence or absence of sperm, is all done in those F2s. The fact that we see variability in the crosses is like we're swapping the different alleles that might be present are from each of those strains. So the F2s, we're not actually like looking. The males that are sterile or fertile are not. How do I say that? Their having sperm doesn't matter on which female we pair the F2 with. He either does or does not have sperm, because we can just dissect the male and he either does or does not have sperm, and it doesn't matter who we made it to. I don't know if that, maybe I'm a little confused on the question.
I guess sorry.
The grid was about which two strains gave rise to that F2 males in question. We were not varying the females involved, that the F2 males were tested against.
But you get these guys like back across to the original population and don't see any problem with fertility.
It's not, they're not, the back cross is what generates the F2 in the other.
Oh, okay, sorry. I got you.
So it's a.
Okay.
Yeah.
Yeah, okay, sorry, yeah.
I don't know if this came through, but the main interpretation we came up with for the back cross result, why wouldn't you get elevated sterility in back cross males in one direction or the other? Was the idea that you might need at least one additional locus, from the same parent as the uniparental factor, and the cross design did not allow that third locus to also be homozygous.
I think, yeah, maybe we can do one more quick question.
Just like super quick.
Yeah, yeah.
So I love the system and I love how reduced it is and how like it's a really clean set of experiments you're doing. So do you think that the sterility is itself the phenotype, or is this potentially pleiotropic epistasis that you're seeing, and then this is kind of an edge? Like, is the sterility necessarily the key thing? Are there other defects that you're seeing that you're choosing to, when you have to focus on something, you could go forever with this product, right?
Right, yeah, so the question is sterility like the end all be all trait to look at here, or something more specific, some of the different things we're seeing? That's a good question. It was the first thing to look at because it's the broadest. Once we started seeing different phenotypes, that opens the door to like exploring those specifically a little bit further. And in recollecting the data, I'm hoping to actually try and collect a little bit more than we would have power to have specifically signed certain phenotypes with in the first place. So I'm kind of narrowing our search a little bit. I think we might be able to ask that question a little bit better. There are also things, so I didn't talk about the behavior a whole lot. We did a lot of behavioral things, and there's a lot of different behavioral components you can examine too in the mating. The way that we are selecting for sterility might not be the best way to look at if there are behavioral problems in some of these F2s, because we're just checking whether they're able to produce offspring or not. So you could have sort of incompatibilities where it makes the males less likely to mate, but that's not necessarily going to show up in our assay for whether or not they're ever able to reproduce. So there are certain traits like that that would be interesting to look at. You'd need a different assay than the one we had for are they able to reproduce or not. But that could certainly be a really interesting thing to look at too. And we maybe have some evidence to point that those are relevant if we went to the effort of digging into them. Yeah.
And then when in your genome scanning, you said that you were looking at heterozygosity, but you're looking for epistatic effects, is what I would think. You're looking for combinations of loci, right?
Yes. Yeah. So we assign ancestry as either being homozygous for each or heterozygous, and then we take any number that we want to compare to each other. Scan for that epistasis.
Really small effect sizes from, yeah, so okay, interesting.
Yeah.
Thanks again, Myron.
Thanks.